Hi guys and welcome back to another video. It's Dr. Nasser here, and today we are looking at a classical peripheral blood smear histological slide. Now, unlike most histological slides that use hematoxylin and H&E staining, blood smears typically stain with Wright stain. With this stain, nuclei appear bluish and cytoplasm or other structures appear pink or reddish. A blood smear allows us to examine different types of blood cells circulating in the bloodstream. The most common cells in the blood smear are red blood cells, also called erythrocytes. These cells are small, dark and pink, have a central pale area, and do not contain a nucleus. Red blood cells are extremely abundant, with approximately 5 million cells per cubic millimeter of blood. They are about 7 micrometers in diameter and responsible for transporting oxygen throughout the body. White blood cells, or leukocytes, are much less common than red blood cells. Typically, there are only about 5,000 to 10,000 white blood cells per cubic millimeter of blood. Several different types of white blood cells can be identified in a blood smear. The most common white blood cell is the neutrophil. Now, neutrophils are so-called polymorphonuclear leukocytes, or PMNs, because their nucleus has multiple lobes. The average neutrophil nucleus has about 3 to 4 lobes. These cells play a key defense role in defense against bacterial infections. Another common white blood cell is the lymphocyte. Now, lymphocytes have a large round nucleus, very little visible cytoplasm. They are slightly larger than the red blood cells, typically measuring around 10 micrometers in diameter. Lymphocytes are important for immune responses and antibody production. Monocytes are larger cells with convoluted or kidney-shaped nucleus. They are the third most common white blood cell in circulation. Monocytes eventually develop into macrophages, which help remove pathogens and cellular debris. Eosinophils are identified by a two-lobe nucleus and also having large reddish granules in the cytoplasm. These cells are involved in allergic reactions and parasitic infections. Basophils are the least common of the white blood cell. They contain large dark granules that may partially obscure the nucleus. These granules contain substances such as histamine, heparin, and serotonin. Basophils play a role in inflammatory and allergic responses. Scattered throughout the smear are platelets. Platelets appear as small purple fragments rather than full cells. They are important for blood clotting and typically present at concentrations of around 150,000 to 400,000 per cubic millimeter of blood. When analyzing a blood smear, it is important to examine well-preserved cells. Cells that are damaged, smeared, or incomplete should not be used for identification, as they may lead to incorrect conclusions. In peripheral blood smear, the main components to recognize are red blood cells, which are the most abundant cells, white blood cells, including neutrophils, lymphocytes, monocytes, eosinophils, and basophils, and platelets, which are small fragments involved in clotting. Together, these elements provide valuable information about blood function and immune health. I hope that was helpful. If you like this, please give it a thumbs up and a comment, and I'll see you in the next one.